introduce, my name is Joy Johnson and I work as one of the senior BMSs at Southampton Hospital Blood Transfusion. And as part of my master's project, I did a study on the relationship between the outcome of the SARS-CoV-2 virus, known as popularly as COVID-19 infection, and patient blood group. And, I'm just going to share that as we go along. Next slide, please. Sorry, next slide, please.
I've moved to the next slide.
Oh, mine's not moved. Okay, that's fine. If you just let me know what slide you're on, I've got the presentation on a different computer, so I'm just going to talk you through, but my slides are not moving.
Okay.
Okay. So
content.
Yeah, so content now. So what I'm just going to talk about a bit about the emergence of SARS-CoV-2, and don't worry, it's not going to be a virology class. A bit on blood groups and disease association, the aims of the study that I did, the method of how, what I did in terms of data collection, how I analyzed my results, and a bit on limitations and conclusion. Next slide, please.
The emergence of SARS.
Thank you. So with the first slide, you can see a bunch of people
yes,
huddled together, and that's what we're used to, everyone carrying on live, doing things together. And if you click next, please, you notice then the rule for the 2 meter distance, everyone had to be apart. And if you click the next, please, you would see some people with a face mask, which is the current situation as of date as well. So how did we kind of get from, this cluster to the fact that we have to wear face masks and we have to, sit apart each other or stand apart each other? So as I was explaining before, I don't know which part I cut off initially, but what started as a pneumonia of unknown origin then happened to spread, quite quickly all over the globe, and everyone heard word about it, and of course the WHO then declared a global emergency. So of course the race was on to find out, what the virus is, basically how do we deal with this, and it was named the SARS-CoV-2 virus. And of course, we're not novel to the coronaviridae family of viruses. And the picture on the right, which I hope everyone can see, just gives the structure of the SARS-CoV-2. So as we all know, as an RNA, it's single-stranded RNA. And on the membrane of the virus, you've got the spikes as labeled on there. And if we magnify into one of the spikes, you can see it's got a receptor binding domain, which binds to the ACE2 enzyme receptor of the host cell that the virus will infect. And then, of course, the viral RNA then goes into the host cell and causes the infection, basically. So that's how the infection happens. So if we go to the next slide, please. Okay.
Global
so
pandemic.
As I mentioned, thank you. As I mentioned before, of course, so these were the recent numbers as of the last time I checked. But of course, we noticed the numbers were going up. It was spreading quite rapidly, around the globe. And just to, it's not news to anyone how much, how high the infection is or how infectious it is, or, the implication of the infection so far. So this is just the data of the WHO website. If we go to the next slide, please.
Risk factors.
Yeah, okay, thank you. So, of course, with the alarming numbers, somebody had to do something. We couldn't just let it keep going. And, especially with the mortality rates increasing, there had to be extensive research into the epidemiology, the pathogenesis of the infection. And, of course, various studies were being put out there to look at, what could be causing the virus, what are the risk factors people should be aware of, and basically what was influencing the virulency and where was it spreading rapidly. So I was just going to share quickly. So one of the studies that were done, especially because, like I said, the coronaviruses, they're not new to the world. When the SARS-CoV-1 happened, it started off in China, spread to about 24 countries. There was 813 deaths. And I'm pretty sure when this one started as well, they thought it would be similar, not expecting it to go as massively as it did. Some of the studies that looked at the first coronavirus found that people who had who were blood group O, they tend to be less susceptible to infection or the consequences of the infection. So there were studies thinking, maybe we could look at, how the blood group is affected in COVID infections. So some of the risk factors that were brought up from all these studies included age. We saw in the media as well that the elderly populations were being affected more, being hospitalized more, for different reasons, but also with a positive COVID result. We heard a bit about gender. It was affecting the male population more. Of course, people with comorbidities tend to have worse outcome when it comes to secondary infections. And if you click, please. Yeah, there was also suggestions of maybe ethnicity, even though I couldn't really find a paper to support that, support a reason for that, and also blood group. If we go to the next slide, please.
What if?
Okay. Just join. Okay. So a bit of a bright idea, like I've mentioned before, it's not the first time the blood groups have been associated with, the prevalence and outcomes of various diseases. We know with VTE, patients with blood group O tend to have better outcome because they have less factor VIII and less von Willebrand's. But the problem with that, when it gets to major hemorrhages, they tend to bleed more. We've seen the blood group implication with malaria, with individuals with the Duffy antigens and the red cells, how they, because it uses the Duffy antigen to infect the red cell. So what I call it natural selection did was people were losing the Duffy A and Duffy B antigens. With influenza and respiratory diseases as well, we see that as I mentioned before with the SARS-CoV-1 virus, it was thought that the patients who were blood group O, because they had the anti-A and anti-B antibodies in their plasma. So remember the first picture I showed where you've got the spike proteins and sorry, I'm just going to dismiss that. You've got the spike proteins, and the spike proteins tend to bind to the ACE2 receptors. So what they found was the anti-A and anti-B antibodies did bind to the spike proteins, meaning they couldn't bind to the ACE2 receptors on the red cells, so conferring some sort of, resistance to the infection. So if we go to the next slide, please.
Extensive studies.
And if you just click through all of it, please. Yeah, just keep clicking, and one more. Thank you. So, of course, everyone started looking to see, what could be the association with blood group and COVID-19, if there was a link between, blood group and COVID-19, was the RH status of, would the RH status of a patient confer resistance to COVID-19 and all of that. I've just put those studies there just to show that, it was actual, work being done by different groups of people around the globe. If we go to the next slide, please. Okay, so because this was quite rampant when I started my research project, I decided to look at the local hospital which I work in. I was able to get some data of positive patient of the trust, and I wanted to see if there was a relationship between COVID-19 and the blood group. So this slide just basically shows the process of me getting my data and interpreting my results. So firstly, a patient test for test positive for COVID. And the way they would test positive, in virology, they have a gene expert analyzer, which uses a PCR process. So, of course, we're looking at viral RNA. The RNA gets converted. I'm not a virologist, so I don't really know this in much details. Gets converted to DNA, and then it's amplified, and then they can look into, they can pick out a positive reaction from that. And of course, the next stage is then checking if this patient who's tested positive has got historical blood group, because that's the only way I could compare the positive patient with the blood group. And of course, we're all, most of us work in the blood transfusion lab, so I'm not going to go into the details of column agglutination technique, which is what we use here in Southampton. And the next step was to analyze the data using SPSS software. And I must say, I'm not as I don't work well with statistics, but this was, SPSS is a statistics software. So I just looked at things with, the blood group, the RHD status, the gender, the age, and how it affected the infection or the outcome of COVID-19. And then I interpreted the results, and I'll just share some of those in the next slide, please. Okay, so this just showing a basic blood group distribution within the UK, and it was very important to note this because depending on your blood group distribution, it would affect the number of people who would come up positive. So, for example, if you were in a population where 100% of the population were, blood group O, if you got any positive cases for COVID, they would likely be blood group O. So that will kind of skew the data a bit. So just mentioning the blood group distribution, 44% O blood, 42% A, 10% B, and 4% AB blood group. And with the RHD status, RHD positive 85%, RHD negative 15%. So if we go to the next slide, please. Okay, so when I looked at so this was the basic start of the project, blood group and the prevalence of positive results. As you can see, if you just click next, please, actually. Yeah, as you can see, highlighting the percentages of, of the prevalence of blood groups. So in blood group A, I found it that, so I had a total cohort of 1,022 patients, which is a small sample size, but it was good enough for the project I was doing. So 425 people out of 1,022 were blood group A. So all of these people tested positive for COVID, so which was 41.6%. 4% was from blood group AB, 11.1% blood group B, and 43.3% blood group O. And if we go back to the previous slide, but we don't have to, you would remember that this percentage is sort of match up with what the distribution is with the UK. And if you're not going into details, you can sort of at this point see that there's not really a correlation between the blood groups and COVID infection, because depending on how many people you've got, depending on what they test, what their blood group is, there was no relationship, because if you had more people with blood group O or blood group A, you would see a higher incidence in people with blood group O and blood group A, just because that was the distribution. If we go to the next slide, please. Is this one? Yeah, just keep clicking. Thank you. So, so this is blood group and ICU admissions post-infection. And if I had a clicker, I would point, but just underneath the highlighted bit, you could see that 75.1% of patients who were 75.1% of patients who were blood group A were admitted into ICU, and 42% was the total number of patients within blood group A who were admitted to ICU. And looking through the numbers, so with blood group A, AB, B, O, and the expected, so just to explain what the table is, I know it looks a bit complicated. Number one, where on the left-hand side of the table, is for those who were actually admitted to ICU, and number 0 are those who were not admitted to ICU. And if you compare the data, just looking at the numbers, they are quite similar for the group, the expected count, so 425 patients in total, as we said in previous slide, tested positive for COVID, and out of 425, 106 were admitted, 106 patients with blood group A were admitted, 16 patients with blood group AB were admitted, 34 patients with blood group B, and 106 patients with blood group O. And working out the percentages, you can see that there's really no correlation because it's still given a similar percentage to the UK distribution, if that makes sense. If we go to the next slide, please. So exactly the same with mortality rates. We see that 100, so 1 is for those who actually died and 0 are those, for those who survived. So we could see that 134 patients who were blood group A died, 11 patients blood group AB, 30 blood group B, and 136 blood group O. And going back to looking at the percentage, it is still quite similar to what's the UK distribution is. So far we could see that depending on what your population is would seems to be affecting the incidence of what groups you see at each stage. So as positive reactions in ICU admissions and as mortality rates. So if we go to the next slide, please. Okay, so then because I had the data for the RSD status, I looked at if there was any significant difference between patient who tested positive who were RSD negative and patient who tested positive who were RSD positive. And if we remember the slides with the UK distribution, there was about 85% of the general population are RSD positive, and we've got 82.4% here who were admitted to ICU. And with the RSD negatives, there was 15% of the general population, and we've got about 17.6% who were admitted to ICU. So straight away you could see that there was no significant difference. And one way I worked this out was to see the similarity between the actual count and the expected count. So with my null hypothesis to see if the actual count was different from the expected count, then you would call it a significant difference. But I would just share some numbers that I've got. So the actual count for the RSD negative patient I had was 46, and the expected count was 41.8. So they were fairly similar. For the RSD positive patient, the actual count was 216, and the expected count was 220. So also quite similar. So with that data, basically, you could clearly see that the RSD status did not matter in terms of ICU admissions. Going to mortality rates as well, you could see similar figures, 16.1%, 83.9%. The actual count, I'll just see if I can find that. The actual count was 50. The expected count was 49.6, fairly similar. For the RSD positive patient, actual count was 261, expected count was 261.4. So also fairly similar. And what this just means was that the RSD status did not matter if the patient got COVID, if the patient got admitted to ICU, or if the patient died. If we go to the next slide, please. So I looked at gender and mortality rate as well. In terms of mortality, the female gender was 40.1%, which was 243 out of 1,022, compared to the male gender at 59.9%. So we can see the male gender was slightly higher, but there was still no significant difference. And I know the media did, there was talks or there were studies to show that the male gender were having, there was more prevalence, more admissions, and more death. But from my research, I couldn't find that. If we go to the next slide, please. Just want to quick the data. And of course, with patient age and outcome, I was only able to get a mean age of infection, and that was a 63.72. So I put all the ranges. So I think the youngest was less than 1, and the oldest was up to 100. And the mean age for infection was 63. And of course, we saw that with the elderly population, they were, being admitted more, and there was higher mortality with them as well. If we go to the next slide, please, which I think should be the last slide. Yes, of course, with so with the project with limitation, I'm just going to talk about the limitation and conclusion. So some of the limitations were it was a small sample size. So to put it in perspective, I was only looking at one hospital, even though it was a major trauma trust compared to the whole of the UK or the whole of the whole world. But if we had maybe a larger sample size, we might be able to get more definite results. But from my study, I couldn't find any couldn't find any significant difference. So some of the modifications to the study, which we do, of course, have a larger sample size. And as I mentioned, also the distribution of your blood group in a particular country, because I think studies from Southeast Asia found that patients with the B blood group were more likely to be admitted to ICU, were more likely to be intubated. But if you also look at the distribution of blood group, you will find that there are likely to be more patients with the B blood group compared to the UK. And in terms of what next, I mean, it would be really good if we could, look into more, especially with the anti-A and anti-B, how, because some papers have suggested that the, the higher the anti-A and anti-B levels, like I mentioned before, because they bind to the spike protein, the virus is not able to bind to ACE2 receptors on the surface of the cells, so there's no infection happening. So there could be more studies looking at that. There could be, a higher sample size. Could this help prioritize treatment? So could they be doing blood group checks on every positive patient that comes in to say, if your blood group A, should we should we admit you to ICU? Should we start looking at better treatment because you're at a higher risk of, a worse outcome, if that makes sense. But I've put a question mark at the end there because we're all left with questions. It's always a what if, what happened. We're not really, there's no definitive answer. And, COVID is still ongoing. So unfortunately, I couldn't find anything in my research, but it was a very interesting topic for me. And I thought I'd share that with everyone. So thank you.